All right, this is a special sort of mini lecture on flagellar motility to go along with um, the lab for week 2 of microbiology. This material is largely in chapter 2 in your book. There's a little bit of extra stuff in here, some linked videos that you can watch as well. So bacteria, the most common mechanism of bacterial motility or movement is through flagella. These are the only true rotary engines in biology, and they're fascinating structures that require a huge investment of the cell's resources and energy supply. So they must be really important. This is just an EM image, a colorized EM of a salmonella cell showing a bunch of flagella coming out of it. There might be something like maybe 8 or 10, 12 maybe flagella on this. And you see they're coming out from several places on the surface. Bacteria can have multiple flagella spread around the surface. They can have single flagella. Usually if they're single, they're at a pole, but not always. Some bacteria have a few flagella coming out of the pole. There are terms for this that are described in your textbook. I don't care if you remember them that much, but it's important to recognize that this is a variable trait in bacteria. E. coli, like salmonella, typically has anywhere from 5 to a dozen flagella. As I'll show you in a couple minutes, there are, under special conditions, some gram-negative bacteria actually make hundreds of flagella on their cell surface. Usually their cells are a lot bigger than this, but so there's a lot of variability there. So it kind of looks like sort of a flabby piece of spaghetti here, but in fact, in 3 dimensions under natural conditions and in an aqueous environment, these would be sort of semi-rigid screw propellers that when they rotate are going to push or pull the bacteria through the medium. This is quite distinct from eukaryotic flagella, which typically will sort of whip back and forth like the tail of a sperm. I'm not going to click on this link right now, but you can look on this link and there's some diagrams and explanation of the difference between eukaryotic flagella, which are microtubule based, and bacterial flagella, which are definitely not based on microtubules. Okay, so flagella allow bacteria to move. They allow bacteria to swim, and they can move incredibly fast using these flagella. Multiple body lengths, 10, 20 body lengths per second in some cases. And this would be like a person running like a 100-yard dash at like 60 miles an hour or something, right? So very, very fast. And the reason they have these flagella is to be able to help in accessing nutrients. Basically, they combine movement with the ability to control their movement through chemotaxis to sense gradients in chemical concentrations to move towards things they want to move towards, typically nutrients, or sometimes away from things that are going to be harmful to them. Okay, we'll talk about chemotaxis in a minute. Flagellar motility is also important for bacteria that are pathogens that colonize humans or animals or even plants in some cases. And it's probably because it's connected to that chemotaxis, it helps them find the niche that they want to. But if you knock out the flagellar motility, then often these pathogenic species are not able to colonize their hosts. So it's clearly important for them. Now, I don't want you to think that all bacteria have flagella, because they don't. There are many bacteria that don't have the genes for making flagella. In fact, very few cocci make flagella. The bacteria that make flagella are almost always rods or vibrios or spirochetes. Why that's the case, I don't know, but it's not that all bacteria make flagella, but for the ones that do, it's something that's really important to them. Okay, so this is basically what a flagellum looks like. If this is our cartoon of a whole cell. This one happens to be located at the pole. You can see this sort of whip-like structure, which I told you is actually more of a helical shaped structure stretching out here. This can be, you know, even 2-3 times the length of the cell. So given that this is all made of protein, this is almost all made of a protein called flagellin, you can imagine that that's a lot of protein that the cell has made and exported into that structure. So it's a big investment. Running this, making this flagellum turn also requires proton motive force, right? So if we zoom in here to the base of this structure in the cell envelope, as you see here, there's a rod that's penetrating out from the cytoplasm, basically through the cytoplasmic membrane, through the periplasm, through the cell wall, through the outer membrane. Then we have this hook structure here, and then we start our helical filament. So this is called the basal body, the part that's actually embedded in the cell envelope. And this is really critical for a lot of things because, as you'll see in this video, the motor for the flagellum surrounds the base in the inner membrane, and that's where the protons flow through to cause this to rotate. And then in the end of this here is a specialized protein secretion apparatus through which the various proteins that make up these structures are secreted and then assembled as the structure grows out here. Okay, so what I want to do is show you this animation. You can click on this on your own as well. This is just a couple minutes out, and this is going to show sort of how the flagellum is built. So I'm going to stop sharing for a second here. Click on this animation.
Go to rings YouTube, make up the flagellum.
And now I'm going to, stop this, go back here, and I'm going to share my screen again. Go here. Man, this is like a freaking control room. Okay. Do the full screen and start this. And
your motor, which can be divided into two major parts. The stator, which is attached to the peptidoglycan layer. And
hold on just a second. I'm going to take this back to the beginning. Okay.
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rotobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles.
These are some great dark field images here.
Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell's surface, make up the three parts of the flagella. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts. The stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates.
Just to be clear, this is the peptidoglycan layer in here. This is the inner membrane. This is the outer membrane. This is a gram-negative bacteria. So you can see this part of the motor has this extension up into the peptidoglycan to anchor it in place. As you watch this rotate, you see that the sort of the drive shaft of the flagellum is a silver structure that's attached here, but is penetrating through these two rings, which are also attached solidly to the peptidoglycan to hold them in place. So these are sort of the bushings through which the drive shaft is going, right? And this part down here is rotating by virtue of its interaction with the motor.
The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains 2 MoTB proteins, and therefore also contains 2 of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the MoTB proteins. This binding causes a conformational change in MoT-A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the 2 protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called Fli-F come together in the plasma membrane to form the MS ring.
So it's not so important that you remember the names of these proteins or substructures, but it is important that you get the principle here, which is that the assembly of the flagellum starts in the inner membrane, and then this is going to be sort of the foundation that it's going to build on as we build more in here, and then going out, and then there's going to be a channel, a controlled channel that's going to let the subsequent proteins for later structures go out from this inner membrane foundation.
The Fli-G proteins assemble under the MS ring. Fli-G, along with Fli-M and Fli-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. MoT-A and MoT-B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but MoT-B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. The cap rotates, which causes the subunits to build in a helical fashion. A complete...
Okay, I'm going to stop sharing now. I don't know why it's all of a sudden showing all these advertisements on the screen, but I'm going to go back to sharing the PowerPoint. There's not much of the video left. You can see essentially the growth of the filament outward. So what that was showing all was starting here, then the structure growing outward here to make this. I mean, this is really a fascinating example of developmental biology on sort of the nano scale. This is a nano machine that's being built in a bacterial cell with many different proteins. It takes about 40 to 50 different genes to actually make this structure here and the accompanying motor to run it. So it's a very sophisticated structure. I'm not going to talk much about this in this lecture, but it has been shown that there are evolutionary connections between how the flagellum is built and how another structure is built in bacterial cells that is used to inject virulence factors into other cells, particularly host cells like human macrophages. So there are evolutionary connections between building a flagellum and virulence, other than the fact that motility is needed. So that's all I'm going to say about that for now. Let's go on to the next one. So let's spend the last few minutes of this mini lecture talking about chemotaxis, right? So building a flagellum and moving probably wouldn't be much good if the cell couldn't control where it moved somehow. So if it couldn't do useful work with it, if it was just moving randomly in an environment, it's hard to imagine that that would be worth the investment of energy and resources necessary in order to make a flagellum. But chemotaxis is the ability to control movement in response to a chemical gradient in the environment. And you can imagine that the chemical gradient that the cell might want to respond to might be something like the presence of amino acids that they could eat for food, or the presence of sugars, that sort of thing. It could also be the presence of things that the bacteria would want to run away from, like alcohols or organic acids or something like that, that's going to be harmful to the cell potentially, right? So the idea is that the bacterial cell controls the direction of rotation of the flagellum. If you have multiple flagellum, what you find is that only in one direction of rotation do all the flagella form a coherent bundle that allows the cell to swim effectively. In the other direction of rotation, it turns out that the flagellar filaments all fly apart and the cell just spins randomly in a process we call tumbling. Now, if a bacteria only had one polar flagellum, then in one direction of rotation, it would go this way, and the other direction, it would go that way, right? But this is a case when you have multiple flagella, you can get runs and tumbles. Should also point out that bacteria, because they're small enough on the nanoscale, they are also subject to Brownian motion. So just the random vibrational energy of water molecules is enough to actually cause them to sort of jerk around and potentially reorient. So we're not just depending on the flagellum rotation to cause them to tumble and move off in a new direction. But this is, in the next slide here, you can see what chemotaxis is actually predicated on, which is that when a bacteria is moving in a direction that it wants to move, in other words, think of it having a sensor here for whatever chemical is serving as an attractant. If it senses that the concentration of attractant gets higher, then it keeps going in that direction by running for longer, by suppressing tumbling. If it senses that it is going away from the direction of increasing attractant concentration, it will actually tumble more frequently so that it will change direction. So the idea is that between the two of those factors, you get what's called a biased random walk, where if you're going the right direction, you go longer that way. If you're going the wrong direction, you go shorter that way and you reorient. Now, if it's a repellent that's being sensed, it would be just the opposite. If you were going away from it, you would go longer. If you're going towards it, you would go shorter. Now, obviously, this requires having a sensory system, and that's sort of beyond the scope of the current lecture. We're going to talk more about that when we get into signal transduction in the middle part of the course, the mechanism by which the bacteria sense this. But let's go on to the next slide. One curiosity of flagella is that they're not always on the outside of the cell. In the case of spirochetes, the flagella actually are in the periplasm. Spirochetes are gram-negative bacteria, so they have, an inner membrane and an outer membrane, and in between the cell wall, it turns out they also have flagella in there. So if we cut through a cross-section of a spirochete, you would actually see the flagella in this embedded in the periplasm there. So when those flagella rotate, they actually make the spirochete cells rotate. And there are some links in the W2 worksheet for lab that you can actually see dark field microscopic images of spirochetes rotating. It turns out that that's probably useful for a much more viscous environment like mucosal surfaces that spirochetes, at least some pathogenic spirochetes, operate in. A lot of spirochetes are also found in anaerobic environments like sediments, and so they, these are even more viscous than an aquatic environment. The last thing I want to show you is swarming motility. So swarming is a term that's used to refer to using flagella to actually push you along on a solid surface. And this is actually very poorly understood. It requires being able to sense the solid surface and then crank up flagellar gene production or flagellar gene expression to make a lot more flagella. This is a bacteria called Proteus mirabilis, which is really famous for swarming. I actually showed you a picture of a plate of Proteus mirabilis in the week 1 lab. So hyperflagellated just means lots and lots of flagella on the surface. So I'm going to show you this real quick so you can actually see a time-lapse image of Proteus. So I'm going to stop sharing. I'm going to start sharing again. I'm going to go out here to YouTube. Go here, I'm going to go back to the start of this and do full screen. So these 2 spots of Proteus mirabilis were inoculated on the surface of the plate, and then these images are taken over. I'm not sure what this time scale is. There's not much information about this, but you can see the bacteria actually spreading out from the inoculation point across the surface. And for whatever reason, they kind of do this in waves, right? And the edges of these waves are actually dedifferentiated swarmer cells. So you see within about 8 hours, these bacteria have spread all the way across the surface of this plate, okay? So that is swarming motility, and that's thought to have a role in the virulence of Proteus mirabilis. I'm going to stop sharing there and go back to sharing the PowerPoint presentation here. So Proteus mirabilis is a fairly common urinary tract pathogen, not as common as E. coli, but it's still fairly common. We isolated, I think, 4 cases of them in the last year and a half at Santa Clara, right? And it's been shown that in a mouse model, if you knock out the ability to form swarmer cells, then Proteus cannot infect the urinary tract in mice. And so it seems to be really important for moving along those mucosal surfaces. Okay, so that's our introduction to flagellar motility. And I'm going to stop there.